Thank you very much. I want to say that my colleagues in previous discussions did a great job and helped me in presenting the topic, because what we have learned here is that environmental factors are important, genetic factors are important, and Olaf also presented very important data showing the role of inflammation. I think that the most scientific data now we have strongly confirm us that multiple sclerosis is a inflammatory, demyelinating central nervous system disease with secondary neurodegeneration. Of course, here's a simplified picture, but it tells us that in what we believe, all things start not inside the central nervous system. However, there are some suggestions for that, but in outside the central nervous system, in the blood, when we, due to environmental factors and in susceptible individuals, have a process of creating autoreactive T cells, which then cross the blood-brain barrier, go inside the central nervous system. Here they are reactivated with the antigen-presenting cells, dendrocyte and other cells, and produce several cytokines, the enormous amount of cytokines we detect with different functions. Among these functions we have the activation of endothelial cells, we have the activation of astrocytes, which release important molecules. We don't have time much to discuss them, but we know about different chemokines which have different roles. They recruit additional cells. They can activate macroglia cells, which are very important in the process of the pathogenesis of MS. They make the conduction block, and they also can damage the axons. We have two arms in the whole process of the MS pathology, in the relapsing remitting and then secondary progressive. In adaptive immunity we call, which the mainly role of the adaptive T cells and B cells, and then innate immunity with macroglia, monocytes, and dendritic cells. I think that all these arms act together and contribute to the inflammatory and immunological processes which we see in MS, which leads to the structure of the cells, the myelin, the axons, and also other types of cells. Here is I think, one of the crucial also process in MS pathology, in inflammation, when we have the migration of the cells, inflammatory cells. They are multi-step process, including rolling, activation, adhesion, when we know that the integrins on the surface of lymphocytes and also the molecules on endothelial cells like ICAM, VCAM1 participate. Then the protrusion and transmigration process through the also basement membrane with the involvement of metalloproteases. So many molecules which belong to the whole inflammatory process are involved in this important process of migration of cells. We can measure, of course, these molecules, and here you see in the CSF and in the serum that these molecules here, selectins, which are involved in many processes, also in this process of rolling of lymphocytes on the endothelial cells, are elevated. Also the adhesion molecules, soluble forms of adhesion molecules are increased in the serum and CSF, and they are correlated with what we see in MRI in the form of the gadolinium-enhancing lesions. Here you see how many, and not all, chemokines are involved, and look that these chemokines, as I mentioned, play an important role in activation of integrins. They are important in the locomotion process of lymphocytes on endothelial cells, and also in the transmigration, of course, according to the hemotactic gradient. And one chemokine can act on different type of cells. Here you see the cells and the chemokine receptors and chemokines involved in migration process of a particular cell. But one cell can be activated also by different chemokines. I would like also to underline the role of CXL13, which attract B lymphocytes, and the role of B lymphocytes seems to also be very important together with the CD4 or CD8 cells. Here is also the picture which show you the types of effector cells which contribute to the process of the destruction of the particular cells in the CNS, and also, which was not mentioned, the important role of the T regulatory cells, which are also the process of the whole autoimmune phenomenon in multiple sclerosis. I don't have much time to discuss this particularly, but here you see how many cells and how many molecules of inflammatory nature and immunological nature are involved. Here is the classical paper and result by Trapp, who presented the axonal destructure, because the problem is neurodegeneration independent or related to the inflammation. And here you see the pictures showing you the acute axonal transection with the ovoids presented here in the green color, and he showed that there is a correlation between the acute transection in form of ovoids and the inflammatory cells like macrophages or macroglia cells. So these types of reactions are correlated, of course. And another interesting results came from the Lastman group, who also showed not only in relapsing remitting form of the disease, but also in secondary and primary progressive type of the disease, the correlation between the markers of axonal destruction, like amyloid precursor protein you see on the first picture, and the number of the CD4 T cells. And on the second picture, also the correlation between the synaptophysin, another marker, and the T cells who stand for HLA-D. That means mainly macrophages and macroglia cells. How inflammatory process can destruct axons? Well, there are many ways to do it. Of course, the macrophages can be involved. but also the cytokines, the proteases, and also the reaction of the oxygen stress, which is going on, and free radicals also has an important role, I think, in the destruction of the axons. So it seems that these processes are very closely related, and it is not just a simplified reaction of the cells for the damage. Well, it was already mentioned by all of that natalizumab is also one of the, well, say, arguments, natalizumab reaction and the method of action. As you maybe remember, this is a monoclonal antibody which acts on the very late antigen 4, which is present on the lymphocytes, and prevents, to some extent, the migration of lymphocytes, which I was talking about, to the central nervous system. So it actually inhibits the inflammatory reaction, the transmigration of these lymphocytes, and I will not talk too much about the clinical effects, but just I mentioned that it resulted in the clinical effects in form of the reduction of the number of relapses and also on disability progression. So the stopping of this reaction going migration of lymphocytes to the central nervous system also resulted in the disability, I mean the reduction of the disability measured by that way. And B cells, which are also very important cells in the process, not only in form of the production of antibodies, which can distract the myelin, but also as antigen-presenting cells, and they are more efficient presenting cells, for instance, in comparison with dendritic cells. So important cells, and nothing strange that much attention was put in recent years on the ocrelizumab, which is the monoclonal antibody against CD20, which is present on several subtypes of B cells, preventing the B cell reactivation or long-term immune memory. Ocrelizumab is another, I think, important factor to consider this problem, because the ocrelizumab action also resulted in the clinical activity, as you see here. It similarly to natalizumab reduced the relapse rate, had effect on disability, and also on MRI activity in form of the reduction of gadolinium enhancing lesions, which are, as you know, markers of inflammation detected on MRI. It was the comparison with interferon beta 1a given in subcutaneously, so also quite important drug, and ocrelizumab was much more efficient in that way. And the last topic, which was also mentioned by all of the Fingolimod action. Fingolimod is a sphingosine 1-phosphate receptor modulator, and he prevents lymphocytes from the egress from lymph nodes, and by that way, in the end, the migration of lymphocytes to the central nervous system. And again, similarly to the previous drugs I mentioned, which stopped the inflammatory reaction, because these drugs are mainly anti-inflammatory drugs, resulted in also clinical results in form of reduction of relapses, which we want, and also had an effect on disability progression and also on the activity, what we see on MRI. Of course, that all does not exclude that primary degenerative process cannot exist. There are some data which try to show that this phenomenon exists independently from inflammation, but still I think that the major process is the inflammatory process with secondary degeneration. Sometimes this misunderstanding, you know, some arguments that for the independence from inflammation was that there is very little or no inflammation in the gray matter in primary progressive or secondary progressive MS patients, but later on it appeared that there is a intense inflammation in meninges, and the soluble fractures and inflammatory cells from meninges could have an influence on the demyelination in the cortex, which we know exists not only in white matter but also in gray matter. So there are many things we don't know yet, and of course my position is that there's knowledge what we have now, and we will see in future what will happen, and so we must be always open-minded and not exclude any possibility. Thank you very much.